Hello my dear student, welcome back to my channel Study Zone. So today we discuss about bacteria. Bacteria are two types, gram positive and gram negative. But what are these categories and how we differentiate bacteria in these special two categories? Don't worry about it. In our class today we discuss about in detail bacteria and their types. What are the staining procedure and how we categorize bacteria in different categories like gram positive and gram negative. And what are the bacteria of gram positive and gram negative. So let's start.
Bacteria are small single celled organisms. Bacteria are found almost everywhere on Earth and are vital to the planet ecosystem. Some species can live under extreme conditions of temperature and pressure. So, let's discuss about the bacteria and their type. Bacteria have a cell wall made up of polysaccharides that give them strength and rigidity. This is important since bacteria often experience variation in osmotic pressure due to the different solution they encounter, and it is there cell wall which prevent them from shrinking or swelling. Cell wall which prevent them. Osmosis is the process by which solvent molecule pass through membrane from a less concentrated solution to more concentrated one, equalizing the concentration on either side of the membrane. Nearly all equalizing cell walls have a peptide polysaccharide layer called peptidoglycan, also known as moraine. Peptidoglycan is a polymer made up of sugars and amino acids which forms a kind of mesh. Bacteria can be classified based on their reaction to the gram stain, which identifies them as gram positive or gram negative based on the chemical or physical property of their cell wall. Gram positive bacteria have a thick cell wall which consists of to around 30 layer of peptidoglycan. This cell wall surrounds a monoderm, which is a single plasma membrane. Gram negative bacteria surround have a much thinner cell wall consisting of a single layer of peptidoglycan. This layer of peptidoglycan is sandwiched between two lipid bilayer membrane called diatoms. We can differentiate between gram positive and gram negative bacteria by dyeing them with crystal violet and then washing them with a decolorizing solution. Then an counter stain is added, for example safranin or fruit stain. Gram positive bacteria will retain the crystal violet dye and remain purple, while the gram negative bacteria will be stained pink. Note that gram positive bacteria also pick up the pink color of the counter stain. However, this is not visible when they are dyed with the darker purple color of the crystal violet stain. The reason for these staining differences is due to difference in cell wall structure, which is the chief difference between gram positive and gram negative bacteria. The gram stain detect peptidoglycan and since gram positive bacteria have a thick multi-layer peptidoglycan layer, they retain the crystal violet dye. Gram negative bacteria do not retain the uh, dye for two reasons. They have an outer membrane getting in the way of crystal violet, and they lack peptidoglycan to retain the stain. Although both gram positive and gram negative bacteria can be pathogenic, gram negative bacteria are more resistant to antibodies because of their impenetrable cell wall. Unfortunately, these bacteria also develop resistance more quickly. Not all bacteria can be reliably classified though through gram staining. Acid fast bacteria and gram variable bacteria, for example, do not respond to gram staining. Acid fast bacteria are bacteria whose cell wall retain stain particularly well, although they are not closely related to gram positive bacteria. They can't appear purple after the gram stain test. Gram variable bacteria show a mix of pink and purple.
So my dear student, I hope you will know about bacteria and their types. If someone facing problem in मोड ऑफ इंग्लिश, अगर किसी को इंग्लिश में ये चीज़ें मतलब जो डिटेल टॉपिक है वो समझ नहीं आया तो मैं हिंदी में भी वीडियो क्रिएट करूँगी, सेम वीडियो इन हिंदी ऑल्सो. बने रहिए मेरे चैनल के साथ एंड इफ़ यू लाइक माई वीडियो देन प्लीज़ सब्सक्राइब माई चैनल एंड शेयर माई वीडियो. थैंक यू. मिलते हैं किसी और टॉपिक के साथ. टिल देन बाय बाय. टेक केयर.